So what I'm going to talk about today is an unbiased approach to gain new insights into Alzheimer's risk, and then to apply this approach to the aging brain as well, and then basically to see what is similar and what is different between those. So we found some surprising things, which I'm going to share now. So I'm interested in understanding molecular mechanisms that underlie dementia, mainly Alzheimer's disease, and some work with frontotemporal dementia as well. And similarly, what's underlying mechanisms associated with the aging brain, so different cell types in the brain, and the overlap between those. And that's led me to study microglia and how microglia interact with other cell types, neurons, and as you'll hear about, oligodendrocytes as well. So we've been working on microglia for around 5 to 7 years now, and what we essentially do is, transcriptomics is our base experiments, and we perform those in mouse models. We use them from human pathology tissue. We integrate data from cultured cells, and more recently from iPSC microglia as well. And the overall goal is to be able to build gene co-expression networks, which we hope will be useful for drug discovery purposes, with the overall goal of boosting the beneficial effects of microglia and neurodegeneration. So I'm going to start with a basic introduction to the amyloid cascade. I'm sure you're all aware of this. So I like to do this to kind of fit what we know about Alzheimer's into, a simple framework here, but also to highlight what's not known as well. So the initial version of the amyloid cascade, as you know, was proposed by John Hardy and Alsop more than 30 years ago now. And the way it's described is that familial mutations and mutations affecting APP processing and environmental factors lead to elevations in A-beta. And then over time, this can be a couple of decades or longer, the elevated A-beta deposits into plaques, and then again, over time, leads to conformational changes in tau hyperphosphorylation, tau tangles, and it's these that cause the death of neurons. So this initial model was very neuron-centric, all about the neurons. Neurons produce A-beta, tau, and it's these that kill neurons. So what dramatically changed was the advent of GWAS studies, and those initially started revealing genes expressed by innate immune cells, such as TREM2. And it was basically a surprise and a challenge to integrate those into the amyloid cascade. So that's when I first entered this work together, working with John. We basically wanted to understand at what level of the amyloid cascades the Alzheimer's risk genes, mainly expressed by the innate immune system, are functioning. And the work that I'm about to describe, and also parallel work done by Bart de Strooper's lab, and then also later on some nice mouse studies from David Hansen at Genentech and from David Holtzman lab, basically brings us to the idea now that the Alzheimer's risk genes are acting through microglia to regulate the transition from amyloid to tau pathology. And so the model that we have currently is the Alzheimer's risk genes, and we estimate that there's probably around 200 of them, regulating how quickly you move down this cascade. So if you're lucky and you have a good collection of SNPs, genes, you're going to move down this cascade slowly, and you'll probably live a full long life without having cognitive problems. If you have a bad collection of SNPs, you're going to move down the cascade more quickly, and then you'll start showing signs of dementia in your 70s and 80s. So obviously aging plays a role in this as well, but we don't understand the mechanisms linking these. And we also don't understand mechanisms which underlie, the calendar age of the brain, the chronological age versus the physiological or functional age of the brain as well. So that's another challenge that we've been thinking about, an area where we don't understand enough. So in terms of the hypothesis driving this work, what we're thinking about is we're trying to understand genetic variability in response to cellular damage, and that influences disease risk and then longevity. So the key word here is response. We're looking at how the surrounding cells are responding to pathology, to cellular damage. So in the case of Alzheimer's, it's clear it's A-beta. In terms of aging, it's age-associated changes to cells. And so in terms of thinking about the response, that's opposed to thinking about the cause. So we're not looking at how beta is deposited, but the response to the amyloid itself, because There's a collection of people which actually have quite a lot of amyloid pathology in the brain, but they're effective at counteracting its changes, so it doesn't lead to cognitive deficits in their lifespan. So trying to understand those processes. So in terms of this overarching hypothesis, the smaller questions that we ask ourselves is, can we understand what links together the risk genes that we know about? And if we can understand those links, can we use that to predict new risk genes for Alzheimer's and for related processes such as aging? These GWAS data sets now, as you know, are absolutely enormous. So can we make better use of that data? Can we mine it in new and clever ways? And then does this give us new mechanistic insights into Alzheimer's and related processes? So today I'm going to tell you 3 stories. The first of these is understanding new genetic risk associated with Alzheimer's, and there's a bit of a sub-story to that as well, showing some overlap with COVID-19, at least at the level of 1 gene so far. Then I'll talk about applying that approach to genetic risk associated with the aging brain, and then finally changes in myelination and oligodendrocytes with aging. So the general approach that we use is to take the human GWAS studies, starting with the Alzheimer's one, but now moving on to the aging GWAS, and basically looking at the sub-threshold variants from the GWAS and then filtering those with transcriptional changes, bulk and single-cell RNA-seq from human and mouse models. And collectively, I believe that will help us to predict disease risk better, but also give us new opportunities for drug discovery. So GWAS, I've already talked about those. They've dramatically changed the way that we think about Alzheimer's. Most GWAS studies are still focusing on hits that are significant genome-wide, so more significant than -A. That's dash line here, genes like TREM2. However, GWAS still have limitations, and those are discussed by this nice paper by my collaborator, Valentina Escott-Price. The types of limitations you get to GWAS are if SNPs or genes are pleiotropic, so they're not just involved in Alzheimer's, but they're involved in other processes that people may experience, like diabetes, infectious disease. In that case, those pleiotropic variants, they kind of fall down this y-axis of significance, and they're no longer significant genome-wide. So basically, those people with other issues are less likely to make it into an Alzheimer's GWAS. Another limitation of GWAS is if these variants are in slightly different positions in different people in different populations, again, their significance is going to drop. And then a third limitation is actually the accuracy of the diagnosis. So the last GWAS that came out last year, it had around 750,000 individuals worth of data in there. And from that, it seems like the diagnosis is not purely Alzheimer's. There may be some other dementias in there as well. And you can see that from the genes that are being declared there. So that's also another limitation of GWAS. So what we wanted to do with Valentina was to mine the sub-threshold variant, the variation that you see here between these 2 dashed lines from nominal significance to significance genome-wide to see if that can give us any meaningful information. So that brings me to the first chapter of this talk. And what we basically did, the first iteration of this, which we published a few years ago now, was to filter the sub-threshold variants from the Kunkel Alzheimer's GWAS with transcriptional changes that you see in transgenic mice with amyloid pathology and tau pathology. So I'm not going to talk about the tau pathology, that was in the paper. I'll use the amyloid mice here to illustrate the approach. So we're looking at mice aging from 2 to 18 months of age, and we can clearly measure the amyloid plaques as they develop. And then we took the hippocampi from these animals and performed bulk RNA-seq. That gave us reliable expression for around 15,000 genes, and obviously that's a lot of hits to deal with. And so what we did was we used this bioinformatics tool called network analysis. I'm sure many of you are familiar with that, gene co-expression network analysis. And that pulled out around 1,500 genes, which have an expression profile, which is similar to the amyloid pathology. So you can see this is really nice. TREM2, the known risk gene, has got a very similar pattern of expression to the pathology, as does this other putative gene that we became really interested, the antiviral enzyme OAS1. So we have 1,500 genes which show this expression pattern. and the most connected genes are shown as this network here. And we can see everything that has a red oval around it is a known risk gene for Alzheimer's. So TREM2, CD33, all our favorite genes if we're a microglial biologist, SPI1, ABI3. And we found that basically within these 1,500 genes, there was a significant enrichment of genetic variation from the called Alzheimer's GWAS. And then on top of that, we overlaid the subthreshold variants shown with the blue rectangles here. So now these become significant because ultimately what you're doing for a GWAS is you're correcting for the 30,000 genes that you're studying. And now we've narrowed down the genes that we're interested to 1,500, then some of the subthreshold variants become significant as well. So the types of genes, so these are putative risk genes now for Alzheimer's, the types of genes that we've highlighted, ITSCAM, so also known as CD11B, it's a commonly used marker for microglia. So perhaps not a surprise that any variation within that gene may be contributing to Alzheimer's risk. Another gene is the lysosomal protein, LAPTM5. There's a leukocyte receptor here, LALRA5, and OAS1A, which I'm going to talk about in the next slide. So these genes are mainly expressed by the innate immune system, and we call this the amyloid responsive network. So that's where we're up to. So all of our data is available from these mouse gene expression studies on the Shiny app as well, so you can check your favorite genes later. So I'm going to take a little bit of a detour now just to tell you a little bit about a published study. This is still a really active area of research for us. So I'm going to spend about 5 slides talking about this now. So OAS1 is oligodenylate synthetase 1. It's an enzyme which is traditionally known to be downstream of viral signaling. So it's quite a primitive defense mechanism against viruses. It binds double-stranded RNA, causes a conformational change. It produces oligodenylate, which activates RNase L, and that chops up the viral RNA to make it inactive. So this process releases interferons, and they act through interferon receptors and then JAK-STAT signaling to cause a feed-forward loop and upregulate more interferon-responsive genes, of which OAS is one. So that's fairly clear on the viral side. How this is related to Alzheimer's is not entirely clear, and this is something that we're studying in the lab using cell biology. So the leading hypotheses for how this is related to Alzheimer's are as follows. It's possible that double-stranded nucleic acids released from stressed or dying neurons, potentially from mitochondria, are activating this pathway. And also within the amyloid plaques themselves, nucleic acids can become trapped, and that may be a way of activating this interferon system. So those are still active areas of interest in my group. So everything I've said up until now was a prediction that OAS1 was associated with Alzheimer's. So following the first paper, we collaborated with Kevin Morgan at Nottingham to look at his independent cohort of Alzheimer's patients. And together with him, we genotyped the top SNP that we identified from our computational work, this 454 SNP. And he showed that there was an association of this SNP with Alzheimer's in his cohort, which were around 2,500 people, half of them with Alzheimer's and half control. So we were doing this work in the lead up to the pandemic, and then obviously given what's known about OAS1 with antiviral responses, I guess before the pandemic it was known to be involved in influenza, also in some forms of hepatitis as well. So I guess we were speculating whether it would be involved with SARS as well. And actually, like, there were lots of bad things obviously happened during the pandemic, but I guess one of the highlights was that the science moved really quickly as well. And by the end of 2020, beginning of 2021, the first GWAS came out for severe disease with COVID-19. So these are people requiring mechanical ventilation in the critical care wards. And they identified, so this was the Bailey lab in Edinburgh, they identified 10 interferon-responsive genes, which altered the risk for a person developing severe COVID-19, and OAS1 was one of those genes. So we became really interested in basically comparing and contrasting how cells respond to Alzheimer's pathology versus COVID pathology to see if what we learned from one disease could inform the other. And so what we did was we mapped our top SNPs associated with Alzheimer's, shown in red here, with the top SNPs that came from the COVID studies onto the OAS1 locus. And using the 1,000 genomes data set, we found that these 4 SNPs were generally inherited together in the European population, and that having this particular combination of alleles at the 4 SNPs, ATTA, we predicted would increase risk for both Alzheimer's and for severe COVID-19. And you can see that this is present in about 56% of the European population, so it's quite common. And, you know, there's also always a silver lining to these things as well. The column adjacent here, GCCG, is the protective combination of alleles. So we published our SNPs and this data showing links between COVID and Alzheimer's for OAS1, and subsequently other groups have shown that the SNPs we identified are associated with Alzheimer's in different Alzheimer's populations throughout the world. And also Alison Goat has a work in progress as well, showing that variation with OAS1 is associated with Alzheimer's risks. We have validation from other groups and other data sets as well. So it's not entirely clear what the variants that we've identified are doing yet. I guess through COVID, there's a suggestion that they may be altering splicing of OAS1. Our initial analysis suggested that these variants are associated with decreased expression of OAS1. So from the GTEx data, we see that the risk variants have got lower expression compared to the protective variants in tissues that are relevant for both Alzheimer's and COVID, so blood, cortex, and lung here. Also, an independent way of doing the calculations with Valentina via a GWAS assay showed that lower expression of OAS1 was associated with increased risk for Alzheimer's. So to get more of a molecular grasp in terms of how OAS1 might be functioning, we turned again to the transcriptome, now looking at single-cell RNA-seq. So this is microglia isolated from APP knock-in animals and controls. So this is a reanalysis of my collaborators' data, Carlos Alafrigerio. And this is nice data, you know, single-cell RNA-seq. You can use these trajectory plots so that with X and Y coordinates, you can basically summarize the gene expression of individual cells, and you can separate them by their cellular function. So we ultimately get these simple breakdowns of activation status of microglia into 3 main classes: homeostatic microglia, amyloid responsive or DAM disease associated, or interferon responsive microglia. So we found, we performed network analysis on the single cell RNA-seq data, and we identify this network, which contains OAS1 quite centrally. And you can see it's expressed by these interferon responsive microglia, the yellow and orange cells here. And then, you know, doing statistics in single cell data is tricky, so we use other data sets to basically look at the association of age and to look at other mouse models of Alzheimer's. So this data is from an O'Neill publication where they basically isolate microglia from old mouse brains and young adult mouse brains, and they perform bulk RNA-seq on the microglia. And we found that this collection of genes was increased in the old microglia compared to the young microglia, and similarly in a different mouse model of Alzheimer's. So this is a pre-synonym into APP. Transgenic animals, this module of gene shows increased expression in the amyloid animals compared to the controls. So again, nice dependency on age and pathology here, amyloid pathology. So this is mouse microglia. We've done the same using human microglia. So now this is human microglia isolated from people with sporadic Alzheimer's and mild cognitive impairment. This is Martha Oehler's data. So again, we reanalyzed it by performing network analysis, and we derive a network which looks quite similar to the one that we found in mouse microglia. OAS1 is quite central again within this interferon responsive network. We can use these module of genes and go back into the GTEx data set. So I'm sure most of you are familiar with the GTEx data. where you've basically got gene expression from a variety of tissues from people that vary in age from about 40 years of age to around 95. These people are overtly healthy. There's no obvious cognitive problems or pathology in them. And we saw in that data that this collection of genes showed age dependency as well. So these genes are showing increased expression with age. And so I think this speaks to increased incidence of Alzheimer's with aging, but also increased risk maybe of severe COVID in the older population. At least it's giving us hints, so more work to be done there. And then the reviewers asked us to do something that was more relevant for COVID as well. And so we applied the same approach now to lung macrophages, again, single cell RNA-seq from this nice Nature Medicine paper. And again, network analysis gave us this network, which again is broadly similar to the networks that we saw in microglia in humans and mice. And these collections of genes were increased in people with severe COVID responses compared to modest responses to COVID and healthy individuals. And so the summary here is that this module of genes is driving the response of these innate immune cells here in lung macrophages, but in the previous slides to amyloid pathology in the microglia in the brain. So I'm just going to put together side by side now the microglia from the brain responding to amyloid pathology or to Alzheimer's pathology, and the lung macrophages responding to COVID-19. And it's not a surprise that there's a significant overlap in the genes that are present in these networks. OAS1 is quite central to both, but they share other features as well. So this interferon-responsive gene 15 here is a hub gene in both of those. And I really like to look at the data with this network analysis approach because I think it gives us a lot of clues as to what's driving the cellular response. I think this is also a way of potentially intelligently designing biomarkers that may be useful to track disease progression for a variety of diseases. And I guess this data is also telling us that there's a common response of these innate immune cells to the different pathologies as well. So this work is ongoing. We're trying to understand the role of these collective genes and also individual genes as well. The function of many of these genes are not known. So we're systematically knocking them down in different cellular models to understand how they affect cellular function. So
can I ask you a quick question?
Yeah, go ahead.
So the risk variance, you predicted to decrease OAS1 or?
So to decrease the expression. So basically it's like a reduction of function, a loss of function.
And then in these interferon modules, is OAS1 up or down?
It's up.
So these modules protected, like this response is protective? Or?
So yeah, I think ultimately OAS1 itself is protective unless you have the risk variant. So
that would imply that interferon responsive microglia might be beneficial in a disease.
Yeah, I think it gets quite complicated and we don't fully understand it. So I guess my hypothesis right now is that OAS1 is kind of a little bit of a weird cousin of the interferon family. It's not classically downstream, so it's not part of the interferon response per se. It's also upstream as well as downstream of interferon receptors. I guess some studies are coming out now basically which show that interferon receptors actually accelerate tau pathology, and that kind of goes against what we're seeing here. OAS1 itself is protective, but it's also upstream and downstream of the interferon receptors. So I think it's probably doing other things as well. I'll kind of go on to some of that, but we don't understand it fully. But I think even within this network, there's like a protective arm and a detrimental arm as well. I think classical type 1 interferon signaling is probably making pathology worse. Like, I think that's the current thinking right now, but that's being explored. Like David Holtzman's work, I think, shows that, and also what Will McEwen is doing in Cambridge. So I think OAS1 is doing something slightly different. Go ahead.
Just a very quick question, and naive probably, about other viruses as well. Maybe showing OAS1 as one of the interferon responses.
Yeah, I think OAS1 is going to be responding not just to SARS, but to other viruses as well. I guess there's already evidence that it does from the literature. So, I haven't said this directly, but OAS1 is a pleiotropic gene. It's involved in multiple disease processes, and I think that's why we missed it from the Alzheimer's GWAS previously. I guess this gets really complicated now because it suggests basically that the way that you respond to viruses and other infectious disease during your lifespan also plays into your dementia risk. And so that gets really complicated. That could potentially be by the vasculature, but those are the things that we're thinking about. So yeah, that's a good point. Yeah, go ahead.
A couple of slides back with the association of interferon genes with COVID. Why do you think you didn't see more of a stepwise pattern? Because you've got severe on one level, and then you've got mild and moderate kind of on a similar level, and no significant difference between the two. Whereas I kind of expect to see a stepwise pattern.
So between these two, you mean?
Yeah.
Yeah. So that's a good point. I think basically these people are almost the same genetically. So I think ultimately your genetics plays a large part in how you respond to COVID. I think that's one of the things that we've learned. And the whole thing about vulnerable people is that they're vulnerable genetically. And basically it's a bit like a lottery. You don't know what your genes are until you get the virus, unfortunately. So I think genetically these people are similar, and so you mount quite an effective response. It's just, whether you've got it or not that distinguishes you from here. But I think you're right, there should be some other cellular markers that should be in between the two as you're kind of resolving, interacting with the infection and then resolving it and coming back down to normal, I think. But it's perhaps not this module. So I guess in terms of the cellular function, where we've made good progress so far was to turn to iPSC microglia in collaboration with Jennifer Pocock at UCL, and to basically, in a crude way, to mimic the SNP variants that we found using siRNA to knock down the expression of OAS1. So here you can see in the left panel that we've got good knockdown of OAS1. And for all intents and purposes, these microglia, they seem fairly normal when we look at a variety of markers. Where we did see a difference was in response to interferon gamma, which was one of the markers that went up with COVID infection. And with interferon gamma challenge, we saw that there was an exaggerated production of pro-inflammatory cytokines such as TNF-alpha. So there's a significant increase there, both at the mRNA level and at the protein level as well. So this brings us to the model where OAS1, as well as controlling antiviral signaling, it's also involved in controlling the pro-inflammatory output of innate immune cells. So if you have lower levels of OAS1, you're going to get a stronger pro-inflammatory drive, things like TNF-alpha, and that's going to lead to dementia by a variety of routes. So TNF-alpha, if it's produced chronically, is toxic, it's going to damage your neurons. Shane Little has shown that with the astrocytic phenotype that has been studying, but also in previous work that I'm not going to present today, pro-inflammatory factors also suppress protective signals such as TREM2 as well to increase risk of dementia. So I guess that's the current thinking right now with OAS1, and we're studying that in iPSC cells. So that work was published about 18 months ago now, so there's more data there if you're interested, but I thought I'd highlight that. So now moving on, we're going to apply the same approach to the aging brain and to the cell types as they age. And that's the subject of the preprint that's available now on medRxiv. So in terms of the longevity GWAS, we're using these two studies by Deline and Timmers published just before the pandemic, and they're using three measures of lifespan here. So the first is longevity. So this is surviving to the 90th percentile of life and comparing that to the 60th percentile of life. So in real terms, that speaks to a person surviving to 75 years of age for the 60th percentile compared to 89 years of age for the 90th percentile. So basically we're looking for genetic variation that underlies that gap in 14 years. And these are the types of sample sizes we're looking at, around 11,000 to 25,000. So the first measure is longevity. The second measure is parental lifespan. So obviously this is less direct. This is UK Biobank data, 1 million samples here. And then the third measure they used was lifespan. So that's attaining old age without obvious disease, and around 300 individuals here from UK Biobank. So we used exactly the same approach here. We're looking at sub-threshold variants from the longevity GWAS, and then we're filtering those with transcriptional changes, and we've done that with a variety of data sets. Initially, we did that with bog standard wild type control black 6 animals, same study as before, 2 to 18 months. Again, network analysis to find age-dependent genes. So examples are shown here, the lysosomal protein I showed before, LAPTM5, and this interleukin-33 shows strong age-dependent changes. And network analysis on these identified that the microglial network showed the strongest association with age, which is maybe not a surprise, but it's nice to see that here. And we did the same analysis in the human GTEx data set using the hippocampal expression. Again, the microglial module network showed the strongest association with age as well. And so what we're going to do over the next few slides is to look at a few age-dependent modules of interest and to see if there's an enrichment of variation associated with Alzheimer's and longevity. So we'll start with the Alzheimer's clinical data initially. And we saw in the previous slides that the microglial network was strongly age-dependent, and as you might expect here, genetic variation associated with Alzheimer's was enriched within this collection of genes. It looks quite familiar to those of you that study microglia, TYRO-BP as a hub, TREM2 is here, CD33. Some of the other putative risk genes that I mentioned before, LAPTM5, CD11B is here as well. So, and this is the p-value here, so significant for Alzheimer's risk. So the thing in the tail here is that these individuals, they don't have obvious cognitive problems. They may have early stages of amyloid deposition, but this network is driven mainly by age and maybe early Alzheimer's. But these people don't have over cognitive problems. So maybe that's not a surprise there. What was more of a surprise was in the age-dependent oligodendrocyte network. So here, the p-value in terms of enrichment for Alzheimer's risk was significant, but more modest. I've highlighted some of the sub-threshold genes that become significant now. So we have the adapter molecule SHC4 here, which is downstream of tyrosine kinase signaling. We have this RNA processing protein and a mechanosensitive ion channel. This figure also illustrates why I love network analysis so much. They kind of really shed new light on genes, which we thought we knew quite a lot about. So in the age-dependent oligodendrocyte network, we see that presenilin-1 is a hub. And at least before this, I would always think about presenilin as its, what is it doing in neurons, whereas now it's actually a hub gene in oligodendrocytes as well. And I guess the hypothesis from this is that presenilin-1 is in some way involved in driving the oligodendrocyte response to aging. So I think there's a lot of work to be done there to understand what presenilin-1 and some of the other genes that I'll talk about in these age-related processes. So another thing in the Alzheimer's field is that we're always conscious about comparing mice and humans as well, mice involving really early stages of disease. And we took this opportunity to compare these age-dependent networks in mice and humans, and we did that with the preservation analysis. So the y-axis here is basically a measure of preservation. The top dashed line is strong preservation, and the bottom dashed line is modest preservation. And again, it's not a surprise, but it's nice to see here that the microglial and the oligodendrocyte network show modest preservation between human and mice in terms of their age-dependent, in terms of the age-dependent microglial network. So I guess, as we would expect, what this is telling us is that while there's a conservation of some core components between human and mice in terms of the microglial response to aging, there are clear differences as well. in some respects, the oligodendrocyte network was slightly more preserved between human and mice because depending on which way around you do the analysis, whether you compare the human networks to mice or if you compare the mice networks to human, the preservation changes slightly here. So I'm not going to talk about this too much, but we looked at the components of these age-dependent networks that were unique to humans or to mice, and then we did biological annotations of those to try and get an insight into what was human-specific in terms of the microglial age response and what was specific to mice. And it seems like the collection of genes which are upregulated only in humans with aging in the microglia are involved more in recruitment of other immune cells, and more specifically T cells. So it's kind of suggesting that there's an interaction between microglia and T cells in the aging brain, which is less apparent in mice. And in mice, where we see age, so the genes that are unique to aging microglia in mice were involved more in antigen presentation. And perhaps what's telling us is that the mouse microglia are better at dealing with the amyloid in terms of presenting the antigens from the plaques. So obviously a lot more work to be done there, but again, this is helping us derive hypotheses and also help to prioritize what genes we should be looking at in these processes. So now we're going to, in the next few slides, look at the single cell RNA-seq networks. So we performed the network analysis, again, in just the control black 6 mice from the Carlo Sala Frigerio data. Again, his data is nice because he does animals at a variety of ages. And with the single cell microglia, we identify a number of modules, and some of them are age dependent. So again, with these trajectory plots, as before, even though now we're just looking at microglial changes with aging, we can divide or we can separate the microglia into their activation states. And again, we've got homeostatic, interferon responsive, and then microglia that look like amyloid responsive, even though there's no amyloid pathology in these mice. So even aging in these animals is driving this network, which looks like an amyloid responsive network. And this looks like an amyloid responsive network because it has ApoE fairly centrally here, and other genes that some of you may recognize, SPI1, there are antigen presentation genes in here as well. So this network of genes, in terms of risk associated with Alzheimer's from the clinical study, so it's close to being significant, but it's not quite, and I guess that speaks to the low resolution of single cell RNA-seq. Here we've got expression for around 5,000 genes, and also this is mice. If we could do that with human, that would be interesting, but we haven't done that yet. And then again, as before, we're looking at the age dependency of these networks of genes. And again, we see in the annual data set that these genes are increased in old compared to young microglia. So that's a nice sanity check as well, and we can do nice statistics there. So now we're looking at a homeostatic network. So in all the modules that we investigated for enrichment of risk associated with longevity, it was only in this homeostatic network that we saw significant enrichment of variation associated with longevity. So the p-value is significant but modest. Again, there are limitations these data sets and the approach as well. So the group of genes in question look like a homeostatic module. So I guess those familiar with these approaches will know that TMEM119 is a hub gene, P12Y12 is a hub gene here as well, known homeostatic markers. CSF1R is here. There's also a few surprises in here as well to keep it interesting. So the Alzheimer's risk gene, ABI3, clusters with the homeostatic cluster, and the lysosomal protein LAPTM5 is here as well. So basically, we don't fully understand this data, but it seems like these genes are controlling whether microglia remain in a homeostatic state or whether they become activated in response to pathology like A beta. This collection of genes is downregulated or decreased in expression in old mice compared to young mice as well. So it shows the opposite pattern of activation. And again, here we're looking at the yellow and orange cells more on the homeostatic arm compared to the amyloid and interferon-responsive. And so finally, we wanted basically a different way of doing the calculations to identify gene variants associated with longevity in the brain. And so again, with Valentina, we performed this GWAS analysis. So again, this is using the gene expression data from the Vitex data set for 20 tissues that are relevant to the brain and blood. And I'm just going to highlight a few interesting points here as well. So we saw a significant hit for caspase 8. So most of you will recognize that for its roles in apoptosis and necroptosis here. So what this is telling us is that allele-dependent changes in gene expression of caspase 8 are associated with increased lifespan. And we found that caspase 8 was actually expressed by homeostatic microglia. So it's part of this network. Again, the GWAS analysis highlights some interesting things. So we have tau here. We have an antisense transcript for Huntington here as well. And so what this is telling us is that gene variation within these genes and low expression of these genes is associated with increased lifespan. And so obviously the variants we're looking at here are not the variants that you get in the familial diseases where you've got early onset neurodegeneration, but these are other variants that might have more subtle effects, most likely through EQTLs that contribute in some way to longevity or associated with lifespan. So that's the cerebellum. If we look at the blood for another example here, so another hit here was FOXO3. So people in the aging field might recognize this. This is a transcription factor. It's downstream of insulin-like signaling, and it's been long studied by people like Cynthia Kenyon and Linda Partridge, whereby lowering insulin-like signaling in worms and flies causes activation of FOXO3, and that activates antioxidative stress mechanisms to contribute to increased lifespan of worms, flies, and it looks like the same is happening in mice as well. So again, this was nice to see this age-associated gene coming out as a significant hit here. And again, we found that FOXO3 was expressed by homeostatic microglia. So these genes showing variants associated with lifespan were expressed more by homeostatic microglia. So with the GWAS, we get around 100 hits altogether. And basically, these were genes that I don't recognize very well. So we wanted to try and understand what pathways they might fit into. So we used ingenuity analysis to try and understand what the connection might be. And about half of those genes, about 50 of them here, seem to be involved in ApoE signaling. And so they're presumably upstream or downstream of ApoE. And so I guess it's not a surprise. ApoE has a big effect on Alzheimer's risk. It also is strongly associated with lifespan as well. And many of the other genes that we've highlighted are presumably contributing to function of ApoE in some way. So caspase 3 is here. A number of cathepsins are here as well. And so again, this helps us derive hypotheses and prioritize genes to study cell culture in mice. And so for the last few minutes now, I'm going to talk about links with aging and myelination as well, because we saw a few surprises there. So we're going to take a few steps back now and look at the age-dependent microglial network in mice. This was the black 6 animals. And again, this network looks microglial to all intents and purposes. ApoE is here. So this is bulk RNA-seq data. So ApoE, it could cluster with any cell type. But here, with the age-dependent aspect, it's clustering with microglial function. The lysosomal protein LAPTM5 was here as well. However, in the center of this module, there were a few surprises. So interleukin 33 and a few of these other hub genes were expressed more by oligodendrocytes than by microglia. And so we wondered if it was basically because there's a close interaction between these cell types and that they're talking to one another during aging, and we wanted to explore that further. And the way that we did that was we used this user data set where they took young control animals, and they treated them with this demyelinating agent, cuprazone. They isolated the microglia, they did RNA-seq. And then what we've done here basically is we perform network analysis there, and we look to see if our age-dependent microglial networks, how they behaved basically in this demyelination data. And you can see here basically that most of the networks that we identified that were age-dependent were also preserved in the demyelination experiment as well. So what this is effectively telling us is that the way that the microglia are responding during aging is similar to the way that microglia are responding to demyelination as well. And so we'll quickly look at two examples here. This was the amyloid responsive network. It increased with aging, and here it's increasing with demyelination. And the homeostatic module, which was associated with variants associated with longevity, was decreasing with aging, and here it's decreasing with demyelination, so basically going in the same direction. And so I'm going to try and bring together, I've told you a lot, try and bring it all together. We've shown that risk genes associated with Alzheimer's, and we've identified around 100 putative risk genes associated with Alzheimer's, are expressed mainly in activated microglia, so microglia responding to amyloid, and to a smaller but significant degree in oligodendrocytes. And the types of genes involved are the genes that we know about, such as TREM2, SBI1, OAS1, and then more putative genes that are yet to be studied, LAPTM5 and this RNA processing gene expressed by oligodendrocytes. And at the opposite end of a life cycle of microglia, homeostatic microglia, and that's where we saw enrichment of variants associated with longevity. Again, we have around 100 genes of interest there, and examples of those are caspase-8, FOXO3, and the cytoskeletal protein AD1. In terms of what's common between longevity and Alzheimer's, with our GWAS data, there was actually very little overlap, and that was limited just to these three genes here. So ApoE is what we might expect. And then the other two that overlapped were this Gamin7 and ZK-SCAN5. So this is an RNA processing gene, and this is a zinc finger protein involved in gene expression. So quite little is known about these two genes, so it's not clear how they relate to Alzheimer's and aging. And so basically, this suggests that we have a two-step model whereby if you have a good collection of SNPs during aging, those lucky people are going to be able to keep their microglia in a homeostatic state and maintain homeostatic processes, and so age successfully. Whereas people with bad SNPs, their microglia are going to be escaping this homeostatic state more readily. And then a different collection of SNPs to genes such as these are going to push some people into dementia. And I guess that's our working hypothesis right now. So I guess the aging field talk about inflammageing quite a lot. And I guess we believe that we've identified some of the gene drivers behind inflammageing, at least in the brain. So we'd like to investigate these in cell culture and in mice as we move forward. So to wrap up, I've said that risk for Alzheimer's is enriched in microglia and some enrichment in oligodendrocytes, showing gene variation associated with longevity is enriched in homeostatic microglia. And finally, I've shown that a substantial part of what microglia are doing during aging, maybe to respond to changes in myelin status. So the paper is on the medRxiv. And I guess our data has a lot of implications going forward as well. So it's really interesting that microglia are responding to myelin as well. So I think that's an area that needs more study. Some groups are already doing some nice work in that area, such as Veronica Miron's group. And I think there's a lot more to be done there now that we have a sense of what genes may be driving this effect. I think there's also a challenge in here as well to develop better markers of myelin status with aging as well. So I don't know if that can be done with imaging, but I think from our data, we may be able to identify some genes which may be good biomarkers to track changes in myelination with age, and that might give us a measure of the physiological age of the brain again, by looking at the genes we've identified, the SNPs, the expressions, and maybe developing immunoassays to measure these. So I'm going to wrap up there. I'm going to thank my mentors, John Hardy, Valentina Escott-Price, Jennifer Pocock. So I guess I'm really proud of these studies as well, because a lot of them took place during the pandemic as well, and they were led by a collection of really great master students that are now going on to do PhD. So Andy Graham was driving the transcriptomic side of things, and he was a really brilliant student. Najia was also brilliant, and she was doing the cell culture with the iPSC work. Effie in Cardiff was doing the GWAS analysis, and they had a lot of help from Umran, Meryl, and Naomi as well from the transcriptomic side as well. And thanks very much to our funders as well. You make this work possible, so thanks very much.